Hello everyone. صباح يا شباب طلاب سنة ثالثة طب. Welcome to the endocrine module. I'll be teaching you endocrine physiology, and this is my first lecture entitled to Introduction to Endocrinology or General Principles of Endocrinology. Before I go further, I'm Professor Zuhair Hassan from the College of Medicine at Hashemite University. I worked previously at Jordan University of Science and Technology and Arabian Gulf University College of Medicine. I'll be here to help you, and I'm glad to help. If you have any question or any query, please do not hesitate to contact me. I will post a channel on Team. I will post my recorded lectures, my PowerPoint slides. I would rather have an online session, live session. It's much better. We can communicate, we can interact. It's unfortunate that we ended up with this coronavirus that put severe restraints, pressure, stress in all of us. We have to manage. But again, I'm here to help you, and I want you to succeed. And I hope we will be able to achieve the mission together. Here are some of the references for you. If you don't like Guyton, can go to Ganong, Review of Medical Physiology, Costanzo Physiology, BRS Physiology, and USMLE. Here, this is an overall view about the control systems of the body. I'm sure you know that we have a couple of control systems, the nervous system and the endocrine system, and these two systems work together basically to coordinate and integrate our body functions. Now, as you may know, the nervous system releases neurotransmitters, whereas the endocrine system releasing hormones. And the name hormone actually came from a Latin word means set in motion. Usually, neurotransmitters are paracrine agents. They just diffuse across the synapses, whereas the classical hormones are circulating hormones where they are carried to their target cells by the blood. The action of the nervous system is rapid, whereas the action of the endocrine system is a bit slower. The hypothalamus and the pituitary gland are the two structures of the two systems which basically related to each other, and that's how through the hypothalamic pituitary axis we can basically coordinate the actions of the nervous system and the endocrine system. This is an overview of endocrine function. I'm sure most of you are familiar with the themes, the main themes of the endocrine system: maintenance of internal environment, sodium, water, balance, blood pressure, blood volume, calcium, iron, phosphate, and name it. It's important for sexual and CNS differentiation in the fetus. It's important for our reproductive functions, important for our development and growth, adaptation, our adaptation to emergency and stress, regulation of energy balance and metabolism, etc. Before we go further, you know, the hormones usually classically were defined as chemicals which are released by an endocrine gland, circulate in the blood, released into the blood, and they are carried to the blood by the blood to their target cells here. And these are known as circulating hormone. Most of our hormones belong to this category. The gland will release after it synthesizes the hormone upon a specific stimulus to release the hormone. The hormone will go through the capillary and then through the blood vessels. It will be carried within the blood to the target tissue, and it interacts with own receptors. And these are the different classes of hormones based on their chemical structure. We have peptide hormones or protein hormones, steroid hormones, amino acid amine hormones, eicosanoid hormones, and this classification basically depends on the structure of the hormone. There is nothing about it. You'll learn more about it in biochemistry. And in this slide here, you have different types of hormones and amino acid derivatives like dopamine, catecholamine, thyroid hormones, small peptides, GnRH, TRH, somatostatin, large proteins hormone like insulin, PTH, growth hormones, steroid hormones such as cortisol, estrogens, androgens, vitamin D, icosanoid hormones include leukotrienes, lymphokines, and prostaglandins as well as leptin. Sometimes, based on their structure, some hormones are lipid soluble, whereas others are water soluble. Water soluble molecules are steroid hormones like estrogen, androgens, thyroid hormones, and usually a large proportion of these hormones, when they are released from the gland, they bind to plasma protein. So they are highly bound because there are plasma proteins in which binds these hormones tightly. They have high affinity for these hormones, and we'll talk about this down the road. And usually these hormones, like the thyroid hormone, interacts with intranuclear receptor. Steroid hormones interact with or act on receptor within the cytoplasm or intracellular receptors because they can easily penetrate the cell membrane. And usually once the hormone interact with its receptor through a series of steps, it stimulates gene expression, messenger RNA, and the protein synthesis. And the protein synthesis and changing enzymatic activity, etc., will induce a response in the cell. And these usually have long half-life and low rate of metabolic clearance, relatively. Now, the water soluble include amine hormones, peptide hormones, icosanoid hormones, everything which is water soluble. These, because they can't cross the cell membrane, they act on membrane receptors. Usually they are stored in vesicle and they are released by exocytosis from these vesicles. And they are mostly dissolved in the plasma freely. They are not bound and they are degraded fast, and that's why they have a short half-life. I just want to mention, compared to the water-soluble molecule which are released by a process of exocytosis, usually thyroid hormones and steroid hormones, which are lipid soluble, are excreted through or released or secreted by diffusion. We'll talk about this later. Sometimes, particularly the peptide hormones, can be classified into families like insulin family, glycoprotein family, pro-immunoglobulin family, secretin glucagon family, growth hormone family, neurohypophysia family. And this classification depends on the site of origin of these hormones. For example, neurohypophysial family, because they are released by the neurohypophysis and include ADH and oxytocin. The glycoprotein family is based basically on their structure because they are a glycoprotein. Insulin-like family, because they have an insulin-like effect, so to speak. They include insulin-like growth factor, and the list go on. I don't think it's terribly that important that you know this type of classification, but it's good to have an idea about it because maybe when you go and read in the literature, you will come across this classification. Now, this slide just shows you the steps of the synthesis and the secretion of the peptide hormones. Let's say this is like a pituitary cell which releases a thyroid stimulating hormone. The hypothalamus will release TRH. TRH will bind to a plasma protein and induces transcription and the messenger RNA formation. This guy will leave the nucleus, go to the ribosomes, and induce the synthesis within the endoplasmic reticulum. And after these hormones are synthesized, they are stored, they will leave the endoplasmic reticulum and bagged into vesicles in the Golgi apparatus, and they will be stored in the vesicles. And once the TRH stimulate the TSH secreting gland cells, it will increase the calcium permeability or cyclic AMP, and this will stimulate the release of these hormones by exocytosis. Just want to mention that once the synthesis of these hormones, they are large and they are biologically inactive, and they are known as prohormones, and they are during the processing and the packaging, etc., they are cleaved to smaller prohormones within the endoplasmic reticulum, and then they are transferred to the Golgi apparatus and bagged into secretory vesicles. And there are enzymes which cleave these hormone into a smaller unit, basically the biologically active hormone and its inactive fragment. And usually once a stimulus causes the release or the secretion of these hormones, they are released by exocytosis into the active hormone and its inactive fragment. I'm sure you are aware of this already. Now we come to the amino acid synthesis and release. The amine hormones are derived from tyrosine, and norepinephrine and epinephrine are released by exocytosis, for example, whereas thyroid hormones are released by diffusion. We'll talk about this. And these are the different glands which release these amine hormones. They are summarized right here. Hypothalamus releases dopamine, thyroid T3, T4, adrenal medulla norepinephrine and epinephrine. Now, the steroid hormone synthesis and release is a bit more complicated, particularly the synthesis. It involves so many enzymes in different layers, for example, in the adrenal cortex. The details of this will be discussed in the biochem lectures. But let's say this is a cell that releases glucocorticoids or cortisol. Now, this cell is usually stimulated by the release of another hormone from the pituitary gland, namely ACTH. Once ACTH is released, because it's a peptide hormone, it binds with the J protein coupled receptor, and this binding will lead to the coupling and the activation of the enzyme adenyl cyclase, which is associated with these G proteins coupled receptors. ATP will be converted to cyclic AMP. Cyclic AMP will activate the protein kinase, and then once the protein kinase is activated, the protein will be phosphorylated, and this phosphorylation will lead to the activation of the cholesterol esterase and the split of cholesterol from the lipid droplets will go to the cytoplasm. The cholesterol, it goes through the mitochondria and through B450 enzymes, along with the shuttling of between the mitochondria and the smooth endoplasmic reticulum. Eventually, we will synthesize cortisol, and the increase in the synthesis apparently increases the concentration of cortisol in the cell, and it will be released by a process of diffusion, or it will be released simply by diffusion. And this just basically summarizes the synthesis and the secretion of steroid hormones for you to read if you can follow up from the previous slide. Let's talk now about how the hormones produce their effects and cause a change in the activity of the cell, or how the hormonal signals is transduced to cause an effect on their target cells. We have these lipid-soluble hormones like steroid or thyroid hormones. because they are lipid soluble, they can easily cross the cell membrane. They go and bind either with cytoplasmic receptors in the case of the steroid hormones, or they could bind with the nuclear receptor in case of the thyroid hormone. The binding of the hormone with its nuclear receptors will eventually lead to the change in the gene expression and transcription of messenger RNA. Messenger RNA will eventually induce the process of protein synthesis like enzymes, and the formation of these enzymes will change the activity of the cell and induce a response in the cell to this specific hormone. At other times, or other types of hormones, they work on membrane receptors, particularly J protein coupled receptors. These include the peptide protein hormones, amine hormones, and these guys, they don't act directly in the protein synthesis. What they do, the J protein couples these hormones once they bind to activate other enzymes, for example, in the cell membrane. So if a hormone, when it binds with the receptor, activate adenyl cyclase, ATP will be converted to cyclic AMP, and this will be a second messenger that will eventually cause protein phosphorylation or dephosphorylation and change in the enzymatic activity, and the cell response will be there. At times, another hormone may activate phospholipase C, which causes the formation of two second messengers, IP3 and diacylglycerol. At times, the binding of the protein with the J protein, the binding of the hormone with the J protein will cause the increase in the entry of calcium. The calcium will interact with another protein known as calmodulin, and this calcium calmodulin complex will induce an effect eventually on the cell. Let's talk a little bit about the J protein coupled receptors and what happens after the hormone binds with these receptors. First of all, these J protein coupled receptors are known because they bind GTP. Now they have three subunits: the alpha subunit, the beta, and the gamma subunit, as you can see here. Once they are inactivated, the alpha subunit usually binds with GDP. When a hormone reaches the target cell and binds itself with these G protein coupled receptors, the first thing that will happen, the receptor will be activated and GDP will be replaced by GTP. Okay, there will be an exchange process where the GDP in the inactive molecule will bind with the alpha subunit, and this will cause to the dissociation of the different subunits, and the alpha subunit is now bound to GTP and it's in active state. Now, once this happens, this complex, the J protein along with this attachment to JTP, will activate membrane-bound enzymes, and these membrane-bound enzymes will lead to the formation or the activation of other enzymes, and these other enzymes will cause the synthesis or the formation of second messengers like cyclic AMP or diacylglycerol IP3. We'll see this in a little bit in the next slide. Now here you can see this binding of GTP to the alpha subunit and the splitting of the three subunits from each other will activate cyclic adenyl cyclase. ATP will convert it to cyclic AMP. Cyclic AMP will activate a protein kinase, dependent kinase, and this apparently will cause phosphorylation of a protein. And once this happens, a cellular response will be there. The effects of cyclic AMP, there are so many. They could lead to synthesis of channels, increased ionic permeability, uh, activation of other enzymes which might be necessary for the hormonal action or its release, and so on and so forth. And you will learn how different hormones, once they activate this second messenger mechanism, is through the J protein coupled receptors, you will know what they do exactly to eventually induce a response. And now one thing I want to say that GTP has to be released. So after it does its action, there is an intrinsic enzyme or intrinsic GTPase activity, which splits the GTP back to GDP plus phosphate. And in this instant, GDP will go back and bind with the alpha subunit, and then there will be like a reconstitution and a reunion to put the receptor back where it was, where the different subunits are hooked to each other, and the GDP is attached to the alpha subunit, and the receptor will be inactivated. And we have to get rid of this cyclic AMP at the end of the response. There is apparently another enzyme which degrades cyclic AMP to its active metabolite, and this enzyme is known as phosphodiesterase. Now, this is basically a summary of the properties of the J protein coupled receptors, and they are known so because they bind GTP binding proteins, and they couple the hormone receptor to adjacent effector molecules, which are the second messengers, like cyclic AMP. J protein couples the hormones to install triphosphate second messenger system. They have intrinsic GTPase activity. They have three subunits, the alpha, beta, and gamma subunit. Usually the alpha, which is important. And when the receptor is not activated, it binds to GDP. Once the hormone binds with the receptor, there will be some structural changes that will lead to the binding of GTP with the alpha subunit. When this happens, this will lead to the formation of the second messenger like cyclic AMP. And just want to point out that we have some G protein, when they are stimulated, they induce a stimulating effect, whereas others will cause an inhibitory effect. So that depends on the action of the enzyme. These are some of the cyclic AMP effects I just talked about: smooth muscle contraction or relaxation, secretion, synthesis of hormones, altering permeability, like for example, the ADH through cyclic AMP mechanism increase the synthesis of water channel, and this will lead to the increase of the permeability of the epithelial cells in the distal tubule and the collecting duct, and thus water reabsorption is increased. Now let's go to the hormones that act through the adenyl cyclase. This might be a nightmare. They are just summarized here. You just look at them. Whenever you study a gland and its hormones, you will be learning about the mechanism of action of each hormone. For example, when we talk about the growth hormone, releasing hormone, and its effect on the pituicytes or somatotrophs, or will tell you that this hormone acts through cyclic AMP mechanism, through the J protein coupled receptor. And so you don't have to memorize them because you will learn them step by step. It is good to have such a nice summary for your exam. Now, this is the coupling mechanism through the phospholipid second messenger system. See the hormone again bind to a J protein coupled receptor, and this enzyme is known as phospholipase C. And once it's activated by the J protein coupled receptor after its binding with the hormone, this enzyme basically breaks down the phospholipid PIP2 or phosphatidyl-indole 4,5-diphosphate. This will cause the formation of indole triphosphate and diacylglycerol. And the IP3 will cause, for example, the release of calcium from the endoplasmic reticulum. Also, IP3 and diacylglycerol particularly activate another kinase known as protein kinase C. Once this is activated, it causes phosphorylation of the protein, and the phosphorylation of the protein will eventually induce a cell response. I am sure the biochemistry people may teach you more than what I am saying here, but it's in your book, so I have to talk about it. This is just a summary of the IP3 mechanism or phospholipid lipase system mechanism. I just summarize them for you here so that you can read them and remember them. There is one thing that I want to point out here, that the diacylglycerol component or the lipid portion of diacylglycerol is arachidonic acid, which is a precursor for the synthesis of local hormones like prostaglandins. Now, this slide summarizes the different hormones which act through the phospholipase system, including angiotensin 2, catecholamines, response to alpha receptor. And remember that the catecholamine, when they bind to beta receptor, they work on the cyclic AMP mechanism. And we have oxytocin here. This is very important because it's one of the important hormones when we speak about milk letdown or squeezing milk from the breast, and it is important for labor. Now, actually, some of the hormones act through a J protein coupled receptor, but this receptor, instead of acting through the ATP cyclic AMP, it acts through a guanylyl cyclase second messenger system. In other words, the second messenger will be formed here from the conversion of GTP to cyclic GMP, and these include nitric oxide, which act as a vasodilator in the smooth muscle, and atrial natriuretic peptide, which plays a role in the regulation of blood volume. I am sure that you have studied these two hormones when you studied the cardiovascular system, so please go and revisit them if you wish. Well, this is another second messenger system known as calcium calmodulin system. If a hormone increases the calcium ion in the inside of the cell or the intracellular calcium concentration, whether by increasing the permeability of the cell membrane and the entry of calcium from the outside to the inside of the cell, or when the hormone causes an increase in the release of the release of calcium ions from the sarcoplasmic reticulum. So whenever we have an increase in calcium ion by one or these two mechanisms, the calcium ion will bind to a protein known as calmodulin and form a calcium ion calmodulin complex. And this guy, the calcium calmodulin complex, activates the myosin light chain kinase. It's an enzyme, and this causes phosphorylation basically of the myosin light chain in the smooth muscle, and the myosin head will interact with actin and cause smooth muscle contraction. Well, apparently, to make life more complicated, we still have other hormones which act through different type of receptors or different types of mechanisms. those receptors which are located in the cell membrane, they are known as receptor tyrosine kinase signaling, or simply tyrosine kinase receptor. And we have two types. One of them, the receptor itself has a kinase activity. So when the hormone binds with the receptor, it will activate this intrinsic kinase, and the intrinsic kinase will cause phosphorylation of proteins, and then eventually it will induce an effect related to the hormone. Another type of tyrosine kinase receptor, they do not have intrinsic kinase activity. However, they are related or non-covalently associated with one type of proteins or different types of proteins known as associated Janus kinases, JAK, just another kinase. Now this JAK has intrinsic activity or tyrosine kinase activity. So obviously, when the hormone binds with this receptor, it will activate the intrinsic kinase activity, which is located within the JAK, or the JAK kinase, so to speak. And now I'll show you these in a minute. We have different shapes, different structures. Some of them are monomers, others are dimers. But the end result we have, as far as the first type, the tyrosine kinases are concerned, one type of receptor is a monomer, and apparently there is at least nerve growth factor working through this receptor. And apparently when the ligand or the hormone binds, it goes to the two monomers, or the two monomers of the receptor, to bind with each other. And this dimerization of the receptor will activate the intrinsic activity of the kinase, phosphorylation of tyrosine moiety on the protein itself and other proteins, leading to a physiological effects. The other type, though, it's already a dimer. I mean, you have two components, but the two components are hooked together. They are attached to each other, and these are the receptors by which insulin, like growth factor and insulin, mediate their effects. So when they activate, when these receptor binds, they activate the intrinsic activity of the kinase and leads to phosphorylation of the kinase itself, the receptor itself, as well as other proteins, and thus we will end up with a physiological effect. I'll show you this in a minute. See, these are the types of receptors, the tyrosine kinase receptors that I was talking about. This is the first type. You can see that it is a monomer, and you see this is the receptor, and here is the tyrosine kinase activity, and it is associated with the receptor. So the receptor itself, the intracellular component of the receptor, has intrinsic activity. So when nerve growth factor, for example, binds, it binds to two monomers, and somehow they are hooked to each other. And once this happens, it will activate the kinase activity that will lead to phosphorylation of protein and eventually a physiological effect of a hormone. Now, the other type, which is also a tyrosine kinase receptor, you can see it's a dimer. The two monomers are connected with each other already, and the insulin will bind with this dimer. And so once it binds, it will stimulate the kinase activity, which is located or which is associated with the receptor itself, and it will lead to insulin effects after protein phosphorylation and enzyme activation, or whatever it is. Now, the other type that I was talking about is the one where the enzyme, there is no enzymatic activity within the receptor itself. It does not have an intrinsic kinase activity. However, it's non-covalently bound with the JAK. It's another kinase, and these are associated with the receptor. They are kinase proteins, but they are, in a sense, they are not the same. I mean, they are not part of the receptor. They are associated or non-covalently bound with the receptor. So when you have a hormone like a growth hormone, it binds with this dimer here, and this binding will activate the JAK receptor, and this JAK receptor will lead to a phosphorylation of proteins, and then it will also cause activation of another type of protein, and it will cause its phosphorylation, known as signal transducing and activating protein. We'll come into this hopefully in a minute. See, that's how insulin acts. This is the tyrosine associated receptor, and this is the tyrosine moiety, or the intrinsic tyrosine kinase here. Insulin will bind. It will cause activation of the tyrosine kinase. The insulin receptor substrate will lead to the phosphorylation of enzymes, and the phosphorylation of enzymes will induce the effect of insulin, like growth, fat synthesis, glycogen synthesis, protein synthesis. It will also cause the formation of a glucose transporter, which are very important for the entry of a glucose to the cells. Okay, now as far as the JAK are concerned, an example of this is the leptin receptor. Here is the leptin, this is the dimer of the molecule, and these are the JAKs. The JAKs, the genus family of kinases, there are so many of them, but let's not worry about this at the present time. Let's say leptin will hook to the receptor, and it will activate these just another kinases, or the JAKs. And the JAKs do two things. They could directly cause activation of enzymes and will lead to a physiological effects, okay? Or alternatively, what's gonna happen, in the JAK will stimulate another protein moiety right here. And this is known signal transducer and activator of transcription. So when the JAK is stimulated by binding of the hormone to the receptor, it will also activate the SAT right there, the signal transducer and activator of transcription. This will lead to transcription, messenger RNA, and transcription, and this will also lead to a physiological effect. I think this is well explained in your book, so I encourage, if you don't understand exactly what it is, you can go and read it. But to summarize, we have a receptor here. It does not have an intrinsic activity for kinases. However, it is linked non-covalently with another type of proteins which have intrinsic kinase activity. And sometimes, to remember them, you can call them JAK or just another kinase. Now these, when the hormone binds with the receptor, it will cause activation of the activity of kinases in these JAK or the JAK kinases. Now the JAK kinases could have an immediate effect or a direct effect by activation of certain enzymes within the cells right there, or alternatively it will still activate another protein known as signal transducer and activator of transcription, and this activates the transcription right in the nucleus and the formation of messenger RNA, transcriptions, and physiological effect. Now the growth hormones, we mentioned most of them. We have a growth hormone, insulin-like growth factor, leptin, and the prolactin. All of these hormones work through the receptor tyrosine kinase signaling. This is the last slide, thank God, about the mechanism of action of hormones. I touched a little bit early on their mechanism of action. Since they are lipid-soluble or lipophilic hormones, they can cross the cell membrane by diffusion from the cell, and then they go to the cytoplasm or to the nucleus. In the case of steroid, I believe they go to a cytoplasmic receptor. The hormone and the receptor will form a dimer, and this dimer will penetrate the nuclear membrane and reaches the DNA molecule, and there is a region within the DNA molecule known as hormone response element that will basically respond to the receptor hormone interaction. We activate gene expression, messenger RNA formation, and eventually we cause synthesis of proteins within the ribosome and the endoplasmic reticulum. In case of the thyroid hormone receptor, it goes to the nucleus directly. It binds with the nuclear receptor, form a dimer, and it will interact with the hormone response element on the DNA molecule, activate gene expression, messenger RNA formation, and the proteins. And once these proteins in either case are formed, they will change the activity of enzymes, among other things, metabolic processes of the cell, and that's how it works. This is just a summary of how steroid hormones or thyroid hormone works, and I explained this. It is just for you to go through it again if you don't remember it from the graph. Now you can see the formation of different proteins through these hormones will lead to the synthesis of, will lead to the activation of physiological actions, and different hormones will activate different enzymes, different actions within the cell. For example, the steroid aldosterone will induce the synthesis of NA channels in the epithelial cells of the renal distal tubule and the collecting duct, and thus it increases sodium reabsorption. When the glucocorticoids are released, they activate the metabolism of the cell. They activate like glycogenolysis, gluconeogenesis, and so on and so forth. So it's really a beautiful system, I have to admit. Now this is much easier, I hope. We talked about now the hormone synthesis, hormone secretion, hormone structure, hormone mechanism of action, and now we have to talk, this is very important physiologically actually, how we control the hormone secretion. We have different mechanisms for control of hormone secretion. A very important mechanism which is basically regulates the hypothalamic pituitary peripheral gland axis is feedback mechanism. That's one of the most important ones, and it works also for other hormones. That's the main mechanism by which hormonal secretion is controlled. And then we have to remember that hormones or hormonal secretion is affected by circadian rhythm. For example, you'll find that some hormones are high during the nighttime, others are high early in the morning, and this circadian rhythm is affected by input to the secretory neurons in the hypothalamus in an area known as the suprachiasmatic nucleus. And then we have what's known as a product mechanism. For example, when a hormone is released and causes a change in the concentration of a certain substance, the increase in the concentration of that substance will go and inhibit the hormonal release. And this is best shown in the next slide right here. You see, this is the first one. We have the feedback mechanisms, and we have the feedback mechanism. We have both. The most common is negative feedback. Let us say that TSH is released from the pituitary gland, and this TSH will go and activate the thyroid hormone release. When the thyroid hormone release is elevated, the hormone itself will go to the anterior pituitary and possibly to the hypothalamus to inhibit the TSH release in the pituitary gland, and thus the activity of the thyroid hormone will decline. At times, these peripheral hormones could also act at the level of the hypothalamus, and specific regulatory mechanisms will be discussed when we take different endocrine glands and different hormones. Now, since we have negative feedback, we have positive feedback. Positive feedback is not common. Here, the release of the hormone will increase the activity of the gland itself. For example, if you take the ovary, which the ovarian activity or hormonal secretion within the ovary, we have actually a couple of hormones, but one hormone is LH. This hormone is released from the pituitary gland, and then it goes to the ovary and stimulates the synthesis of estrogen or beta estradiol. Now, when the beta estradiol is high, it will go to the hypothalamus and the pituitary gland to increase the secretion of LH, and LH will further increase the secretion of estrogen. And this type of feedback occurs during the menstrual cycle. Prior to the day of ovulation, there is an LH surge, and this LH surge will induce further synthesis of estradiol. And this apparently, this is increase, or this large increase in estrogen or estradiol is necessary for the process of ovulation or the release of the ovum from the Graafian follicle. That's something that you will study in further details when you take the reproduction. Now, as far as the product mechanism is concerned, I just talked about, we know, for example, when calcium ions are low, the parathyroid hormone will go to the bone and cause bone resorption or bone release. And then the bone resorption or release of calcium from the bone, this tends to increase the calcium ion level or concentration in the plasma. Once the calcium ion level is increased, it inhibits the PTH secretion. So this is considered to be a sort of feedback, but the feedback is not caused by other hormones or by the hormone that is released by the gland. It is rather caused by what the gland is doing in terms, for example, of ionic concentration like calcium. And you can see a main difference here. In this gland, the parathyroid gland is not controlled by the hypothalamic pituitary axis. It's in its own. And thus, the only thing which is going to stimulate PTH secretion is the change in calcium ion. If we have an increase in calcium ions for one reason or the other, the activity of the parathyroid gland will be suppressed and less PTH will be released by the gland. By the contrary, when we have low calcium ion concentration, it will stimulate the parathyroid gland to release its hormone, parathyroid hormone. This will go to the bony tissue and cause the release of calcium or resorption of bone and the release of calcium, and this tends to increase the calcium ion level in the plasma. And once the calcium ions in the plasma are elevated, it will suppress the secretory activity of the parathyroid gland. Now this slide shows some more details about the negative and the positive feedback. And here in this example, we are using testosterone as an example. Now the hypothalamus will release a hormone known as GnRH. GnRH will stimulate the release of two more hormones, FSH and LH. Now the hormone LH will go to the target gland, in this case the testis. The testis will release testosterone. The testosterone will go to different places, like within the seminiferous tubule, it induces spermatogenesis. It could go to the muscles and increase the muscle mass. That's beyond now. Once the testosterone levels, actually the other substance could do this, known as inhibin, but let's talk about testosterone for right now. When testosterone levels are increased, it will go back to the pituitary gland, suppress the release of FSH and LH by negative feedback. It is possible that also the hormone testosterone will inhibit the release of GnRH from the hypothalamus. So we have this loop known as long loop that inhibits the activity of the thyroid, of the pituitary gland and the hypothalamus through this feedback mechanism. At times, the hormone within the pituitary, which is released by the pituitary itself, it is likely that it will go back to the hypothalamus and inhibit the release of hypothalamic peptide. Like in this case, it's going to be a latch, for example, will go back to the hypothalamus and inhibit the GnRH. In the positive feedback, the best example is basically, well, actually, there are two good examples to remember. One of them is what happens during the menstrual cycle in the female. About the mid of the cycle before ovulation, or at the early onset of the menstrual cycle, the hypothalamus will release GnRH, and GnRH will stimulate the pituitary to secrete FSH and LH, and then the ovaries will release the hormone estrogen. Now, usually when there is an increase in the level of estrogen, the levels of estrogen will suppress the release of LH and FSH from the pituitary gland through negative feedback. That occurs in all hormones except in the female at the mid of the cycle. Okay, the increase in estrogen will sensitize the pituitary response to GnRH, and instead of causing a negative feedback, as you can see here, the increase in the level of estrogen will stimulate the anterior pituitary gland for further secretion of its hormone, and thus we will have more estrogen release. And actually, the increased or the significant increase in the LH from the pituitary gland prior to ovulation or 1 day before ovulation, okay, is an important for the process of ovulation. It's known as LH surge. So the fact that high levels of estrogen, instead of suppressing the activity of the pituitary gland to secrete LH, it causes an increase in LH secretion, LH surge. LH will cause a further increase in estrogen, and this will lead to the rupture of the follicle and the process of ovulation. You will learn about it, just remember it. The same thing here with oxytocin during labor. When at the time of delivery or when the baby is at term, apparently when the head gets down, apparently the dilation of the cervix of the uterus sends signals to the hypothalamus to release oxytocin. And once this process starts, further increase in oxytocin release occurs so that the labor becomes intense and the delivery process gets to be completed. So these are a couple of examples for the positive and negative feedback. Now, this slide shows you the effect of circadian rhythms or hormonal rhythm. What that means, if you measure the hormones at different times of the day, you will find it has a pulsatile release type of nature. If you take a growth hormone, which is here is the plasma concentration in microgram per liter, and this is the time of the day. This is early 8 o'clock in the morning. This is midday. This is about 8 o'clock, and this is about midnight. You can see that when we go to sleep, there is the hormones levels start to increase until it reaches a peak about 3 o'clock in the morning, and it starts to decline. If you take glucocorticoids like cortisol, you can see that if you measure them at 8 o'clock in the morning, they are high, they start to decline, and they are lowest around midnight, and then they start to increase and they get higher early in the morning. And this is important because if you take just one sample to assess the level of the hormone, it's not gonna give you the exact reflection of the level of that hormone. So for those hormones, when you do testing for hormones which shows pulsatile release and shows circadian rhythm, you have to take more than one sample, one like at night and the other one in the morning, so that you can get accurate measurements of these hormones. Now, as you can see, this is just a summary of what I said. The plasma hormone concentration do not necessarily reflect the function of a given endocrine gland. Both cortisol and growth hormone show variations related to the daytime or nighttime, or they are affected by the circadian rhythms. Other factors which may affect the hormone level: sleep deprivation, stress, diseases, night work, sleep patterns. Also, the level of hormone depend on the rate of secretion, rate of metabolism, excretion, and rate of metabolic clearance, circadian rhythm, and so on and so forth. Now, here is some more terminology. Responsiveness is basically how much of the response there is after the hormone it interacts with its target cells, and it's usually expressed in those relationships in which the magnitude of the response is related or correlated with the hormone concentration. As the hormone concentration increases, the response usually increases and then it levels off. Sensitivity, on the other hand, is defined as the hormone concentration that produces 50% of the maximal response. If more hormone is required to produce 50% of the maximal response, then there has been a decrease in the sensitivity and vice versa. And this graph shows you the dose-response relation. This is the response and this is the hormone concentration. As you know, you are measuring the response of a tissue or a cell to a hormone. You can see that when the hormone concentration increases, the response increases and then it reaches a maximum and it levels off. There is something here which is known the AD50. I'm sure that you have studied this when you studied the pharmacology. It is defined as the concentration of the hormone that is needed to produce 50% of the response. If there is a shift to the right, meaning that if the AD50 is increased, then that tells you that the responsiveness to this particular hormone has declined. Now this slide shows you the responsiveness of a normal cell or a tissue, and this is an abnormal one. Now you can see in this experiment there is a decline in the responsiveness, and an increase in the responsiveness will not happen by increasing the concentration of the hormone. Now, decrease in the responsiveness could be due to the decrease in the receptors, number of receptors, or decrease in the number of target tissue. And as you can see, if these two things are absent, no matter how concentrated the hormone is, it's not gonna change the response. Now, this slide shows you the change in the sensitivity. Here is a normal response, and here is the AD50 here. In this dotted line here, which is a decline in the sensitivity, you can see a higher concentration of the hormone is required to produce the same AD50 of a normal response, and the decline in the sensitivity could be due to the decline in the hormone receptor affinity, decreased hormone receptor number, or hormone receptor resistance. There are more terminology here. We have resistance to the hormone. Resistance to hormone, there is some abnormality of the binding of the hormone with the receptor, or it could be there is something wrong with the machinery or the cascade reactions or the cascade events that follows that occurs within the cell due to the activation of the hormone by the receptor. And that's probably what happens in diabetes. There is a hormonal resistance and they do not necessarily respond that well to insulin, or it could be actually both, a decline in the level of insulin. And if you measure the saturation of the receptors, it will be fully saturated. So, I mean, you know, maybe the receptors are there, but their responsiveness is not as good. Now, there is another terminology here known as permissive action of a hormone. Means that the action of a hormone is related to the presence of another hormone. In other words, for one hormone to work, you need another hormone. And a good example of this is, for example, glucagon causes gluconeogenesis, and for glucagon to cause a new gluconeogenesis, you need cortisol. And another example of a permissive action, thyroxine increases responsiveness to catecholamines, because thyroxine, particularly in the heart, because thyroxine increases the synthesis of beta adrenergic receptor, and thus it increases the responsiveness, or it has a permissive action of catecholamines, at least in the heart. Now there is also what's known as downregulation and upregulation, and that what means regulation of receptors affected by the hormone. In other words, the hormone level will determine the sensitivity of the tissue by regulating the number of sensitivity of receptor, the number or the sensitivity. And that's what meant. In other words, the responsiveness or the number or the sensitivity will change with the concentration of the enzyme. Now you have downregulation, means the hormone decreases its affinity of receptors for itself or for another hormone, and it is a mechanism by which the hormone decreases the number or affinity of its receptor in the target tissue. And the purpose of downregulation is basically to prevent the effect of high levels of this hormone for extended periods. So if you have a state where you have a high hormone level, you don't want to have the sensitivity and the number of receptor at a level where you have a bit lower concentration. So if you have a chronic elevation, to prevent the excessive effect of this particular hormone, the hormone itself will decrease the sensitivity of the receptors, and it will also reduce the number of receptors. And that's what's meant by downregulation. And for example, during pregnancy in the uterus, progesterone downregulates its own receptors and the receptors for estrogen. T3, T4 decrease the sensitivity of TRH receptors in the anterior pituitary gland. Similar to downregulation, upregulation, the proposed mechanism of downregulation could be due to an activation of the receptors, inactivation of some of the intracellular protein signaling molecules like cyclic AMP, for example, degradation of receptors by lysosome after their internalization once they bind with the hormone, or it could be caused by a decrease in the synthesis of new receptors. Now, upregulation means the hormone itself increase the number or affinity of its receptors, or for another hormone, it could be due to increased synthesis of receptor, decrease the degradation of existing receptors. And here are some examples. For example, in the ovary, estrogen upregulates its own receptor and the receptors for LH. Prolactin increase or upgrade its own receptors in the breast during lactation, probably. A growth hormone increase the number of receptors in skeletal muscles and liver. Estrogen increase the number in the uterus as well. Increase estrogen upregulates the receptors for LH in the ovary. I just mentioned this. I'm just repeating it apparently. There is a concept that you should just be aware of it. I don't think you should go to the details of how it's done, but experimentally sometimes, or even clinically at times, they can get an idea how fast the hormone is eliminated. And clearance by definition means how much of the plasma is cleared from this particular hormone at per unit time, per minute. So what they do, they usually infuse a radioactive hormone at a constant rate till the radioactivity reach a steady state. And then they calculate the infusion rate, and they take the blood samples and measure the concentration of the hormone or the level of the hormone by their radioimmunoassay, and they use. Now the rate of infusion will be representing the rate of disappearance at this point, and once they calculate them, they will figure out how much is the metabolic clearance of the hormone. The details of the procedure, maybe if you are a researcher in the future, you will learn more about it, and it's good to know it. It's good to know it, but it's just a concept, and it reflects basically how fast the hormone is eliminated from the blood. And so rather than focusing on the methods how to calculate the clearance, I'll leave it for you to go more search, to read more about it, or if you want to go to the graduate school and get a PhD, then you will learn more about it if you need to. But the metabolic, the ways by which hormones are cleared, some hormones are cleared by tissue destruction or metabolic destruction or degradation by the tissue. Once the hormone binds its receptor, sometimes the receptor and the hormone complex will be internalized by endocytosis, and then further metabolization and degradation occurs, and the receptor is recycled. Some hormones are degraded in the liver and eventually will go into the bile. Some hormones are eliminated in urine through the kidney. Now, if you remember, I mentioned this, the rate of metabolic clearance of water-soluble molecules is higher than the rate of clearance of hormone bound to plasma proteins. And it's important to remember that the rate of steroid hormone clearance is decreased if there is a liver disease, and as a result of this, their level in the blood or in the plasma will increase, because these hormones are conjugated mainly in the liver and then cleared in the bile or into the bile. Let's talk a little bit about the transport of the hormones. They could be dissolved in the plasma, and these are free hormones. And some hormones have really high affinity to bind with some plasma protein. I just mentioned this, I just want to refocus it. And if you look at this slide here, we have specific binding proteins for specific hormones like corticosteroid binding globulin. This binds usually cortisol and aldosterone. Thyroxine binding globulin, it binds T3 and T4, thyroxine and tetraiodothyronine. We have another binding protein for sex hormones, binding globulin for testosterone and estrogen. And we have non-specific binding proteins, namely albumin and transerythrin or prealbumin. It binds, like for example, albumin binds thyroxine, most steroids. The transerythrin binds also thyroxine and some of the steroids. And as a result of this binding, actually, it decreases the rate of metabolic clearance. They have a longer half-life, whereas the free hormones, as I said, they are free circulating. Their rate of clearance is high. Their half-life is short, and so on and so forth. Now, this table shows you here, that shows you the percentage of protein binding and of the hormone. If you take thyroid hormones, a large proportion of the thyroid hormones are present in bound with the plasma protein, like 99%. If you go to the steroids, it's a little bit less right here. And for protein hormones, there is very little binding. I mean, very little. It's a small fraction compared to steroids and thyroid hormones. Now you can see the half-life for the thyroid hormones, because the percentage of bound hormone is real high. It could last as 6 days or in days, whereas for glucocorticoids, the half-life is in minutes. And for proteins, it varies from one protein to another. For example, thyrotropin 50 minutes, insulin 8 minutes, antidiuretic hormone 8 minutes. If you take growth hormone, it's about 20 minutes. And you can see here, if the hormone is free and it's not bound, you can see its metabolic clearance usually is high. If you take insulin, it has high rate of metabolic clearance. Antidiuretic hormone has high rate of metabolic clearance. The interesting thing here, the rate of thyrotropin clearance is a little low, and at the same time it's not bound. I really don't know how to explain this. It is just an observation. If you look at the metabolic clearance, how many mLs of plasma is cleared from this hormone per minute, you can see it is very low for thyroid hormone. It gets a little higher for steroids, but still it's a little low. And of course, here the metabolic clearance is not only going to be affected by the percentage of binding, it's how the hormone is degraded as well. And this diagram shows you the relation between the amount of protein which is bound to plasma proteins or to the carrier protein and the free hormone. You can see this is the free hormone. It's affected by the rate of secretion, the rate of degradation, and the degree of binding, and the hormone receptor interaction. Endocrine disorders or other disorders, and one of the ways to do it is to take a blood sample and do assay for these hormones by a method known as radioimmunoassay. And there are several things that you have to remember when you do these measurements. When you take a blood sample, you have to realize that at times for certain hormones, if you take just one sample at any time of the day, or fasting sample may not reflect the true concentration of the hormone, because some of the hormones show pulsatile release or show a fluctuation due to circadian rhythm. So if you just take it, for example, if you take glucocorticoids and measure them in an early morning, in a fasting early morning, you will see that their level is high, and you may get the impression that you have a hyperfunction of the gland. So to have a better estimate, you have to do it one time in the morning and one time in the evening. We just talked about this. At other times, for example, if you take a growth hormone, if you measure this growth hormone level in a fasting plasma sample from a patient, in a plasma sample from a fasted patient, then you may end up with having a low concentration of a growth hormone, and you will end up with saying, well, listen, this person has a low growth hormone level. So you have to worry about these if there are hormones which shows fluctuation with circadian rhythms. You have to assess the overall health of the patient to see if there are liver diseases or something else, so that your measurement, so that you can understand what these measurements or these results mean. And going back to growth hormone, actually we'll see in the next lecture, or in lecture 3, that the growth hormone goes to the liver and induce the formation of another hormone known as insulin-like growth factor 1. And this guy circulates, and it has a long half-life, relatively speaking. And a lot of times measurement of the level of the IGF may give you an idea of the overall levels of growth hormone secretion. It has a long half-life, 20 hours, compared to the half-life of growth hormone. So a lot of times, if you have a patient with suspected acromegaly due to over secretion of a growth hormone, it's better to determine growth hormones at different times of the day, as well as you need to determine as well the levels of IGF, because these last for 20 hours. So their level will not vary from the morning or from at the evening time. Okay. If you go to the thyroid, a lot of times when you go to a doctor, you measure T3, T4, and TSH. These usually the levels of T3 and T4 can be measured at any time, because they have really, for example, T4 has a long half-life that could be as long as 6 days. So random sample of T4 is usually a good estimate of the daily plasma levels of thyroid hormones. Sometimes you can do urine analysis for hormone. That's not, you cannot do it for all hormones. You can do it for catecholamines, steroids as well, HCG and LH. HCG is a very common. Urine analysis of HCG is common for determination of pregnancy, because HCG dramatically increases at the first couple of months during a pregnancy, and actually you can detect it in urine in a week or 2 weeks time. So it is you can just take a urine sample and to confirm a pregnancy, and if you measure HCG by radioimmunoassay, you can confirm the pregnancy. There is an advantage when you take a urine sample here, because a urine sample usually asks the patient to take it over 24 hours, so you can avoid the peaks or the pulses which occurs for hormonal secretion. For example, in Cushing syndrome, when you take a urine sample and measure the free cortisol there, you can eliminate the pulses, the high and lows of the normal circadian rhythm of the hormone. This is a graph shows you what radioimmunoassay is all about. I don't think you should worry too much about the method, but what they do usually, they bring a specific antibody for a specific hormone, and they mix it with a radioactive hormone, a standard hormone, and they take a plasma sample. And now the radioactive hormone in the sample will compete with the standard hormone, the radioactive standard hormone, as well as the hormone in the sample that you are interested in determining its level in your sample, will compete for the antibody. So if the radioactivity in the sample that you take after mixing the standard hormone, the tissue which contains the hormone sample that you are interested in, it means that there is very little of the hormone that you are interested in your sample. However, if the radioactivity declines with time, it means that the concentration of the hormone is high, and most of the antibodies are occupied by the hormone in your sample. So in this case, this is the aldosterone concentration in your sample, and this is the percent of antibody bound with radioactive aldosterone. If there is a high percentage of binding with the radioactive, when you measure the radioactivity, then there is little of your hormone in your sample. If the radioactivity declines, it means that little hormone, the little amount of, most of the antibodies are occupied by your standard hormone. You don't have to worry again about these details unless you want to learn and do some research about it. The same thing with ELISA. This is a much easier, it's cheaper, and here they take a couple of antibodies that will bind with the hormone, and they will bring a third antibody associated with some enzymes, and there are some substrates and chemicals here, and they mix them together, and they determine, there will be a change in the color after the enzymatic reaction occurs. and they measure basically by colorimetric measurements the amount of dye there, and that will give you a reflection of the level or the concentration of that hormone. Now, one of the things that I want to focus on here, the disorders in the endocrine system, one of the important things that you have to make a distinction between a primary versus secondary disorders. A primary disorder, usually there is a problem with the gland itself. It's not somewhere else, or the hyper or hypofunction of the gland. It is because of a disease within the gland itself. For example, excessive secretion by the adrenal gland in Conn syndrome due to adrenal adenoma, excessive secretion of norepinephrine and epinephrine by the adrenal medulla in pheochromocytoma. It's a tumor of the adrenal gland. Decreased thyroid in Hashimoto thyroiditis. This is an autoimmune disease. Reduced ADH secretion due to damage in the hypothalamus or a tumor in the hypothalamus. Now, a secondary disorder again could be hypo or hyperfunction, but here it could, this is basically due to the disorder. If you are interested in a peripheral gland, the disorder could be due to certain problem within the pituitary gland, or it could be due to the presence of a certain tumor in somewhere else. So the disturbance is secondary to a disease of other organ rather than the gland you are interested in. For example, hyperthyroidism may occur in chronic renal failure because hyperthyroidism acts on the kidney tubule to increase calcium reabsorption. Once the kidney is not working, you have constantly low calcium ion levels and thus hypersecretion of parathyroid hormone. In Cushing disease, for example, Cushing disease, there is an increase in the level of cortisol or hypercortisolism, and this is usually caused by adenoma or a tumor secreting ACTH cells in the pituitary gland. Ectopic secretion of ADH due to tumors and cancers, for example. Inappropriate ADH syndrome. Adrenal insufficiency due to for example, panhypopituitarism and the loss of ACTH secretion by the pituitary gland. Pituitary hypothyroidism. In this case, for example, you measure T3 and T4, they are low, and you measure TSH, and it is also low. That means that the problem is probably most likely due to the loss of ATH secretion by the pituitary gland. Now, a lot of times, to determine some abnormalities or to help in the diagnosis of some endocrine abnormalities, we do suppression and stimulating test. And what that means, you do something, either you give a hormone, you give a substrate like glucose, arginine, and depending what you do to determine or to help you confirm your initial diagnosis. And we have two types of tests. Suppression tests, these are usually if you have a hyperfunction of the gland, there is excess release of a specific hormone. You want to do something to suppress or to stop the release of that hormone. For example, if you do glucose tolerance test, you give glucose to the patient and measure the levels of growth hormone, and they don't stop the release of growth hormone. Or what I mean is that if you give a glucose, usually due to hyperglycemia, the growth hormones level will be low. So if you suspect a patient with acromegaly due to hypersecretion of growth hormone, and you give the patient glucose and the growth hormone level is still high, it means that there is a problem with the growth hormone secretion, and it's more likely that's the cause of acromegaly. A lot of times we in adrenal gland, adrenal cortex dysfunction, we do dexamethasone suppression test, and this is a bit more complicated. You have low dose test, you have high dose test. I'm not gonna go to these details, but for example, sometimes you give a low dose to suppress cortisol secretion. So if you give dexamethasone and at a low dose and the cortisol secretion continues or does not stop or does not decrease the level of cortisol, then it means that there is hypersecretion of glucocorticoids due to a problem within the gland. So you do different tests, high doses, low doses. Sometimes if you want to see if you have hyposecretion in the adrenal cortex, you give ACTH challenge test and stimulating it. So the stimulating test, as I said, I just mentioned, are usually used to test hypofunctions, especially for hypothalamic hormone. If you have, for example, somebody who have delayed puberty, and you could try to give pulse style injections of GnRH to stimulate TSH and FSH release from the pituitary gland, and hopefully to restore reproductive function. If this is something that's not related to serious problems within the hypothalamus or the pituitary gland or the gonads themselves, sometimes you inject tropic hormone like ACTH to see the function of the adrenal gland. If you have low levels of glucocorticoids, sometimes the stimulating test, usually if you have somebody who you suspect retarded growth due to low growth hormone secretion, what you can do, you can infuse or inject arginine. Arginine stimulates the release of growth hormone. So if you give arginine and measure the levels of growth hormone before and after arginine, if there is no change, this will help you to confirm that there is a problem with the hormone ADH in this case. And this slide shows you different types of endocrine disorders that may cause hypofunction. I am not going to go to these details. Just have an idea about it. Probably when we talk about different endocrine, we will touch on some of these, diseases and give you an idea about it. But I think it will be more focused when you study pathology. So you just look at it. You don't have to know the details of it. Just keep it in mind for the time being. And by the same token, you can see there are some endocrine disorders which lead to hypersecretion or hyperfunction of the gland, of the endocrine glands, like adenomas, hyperthyroidism, malignant tumors, cancers, ectopic like ACTH, SIADH, and autoimmune diseases like Graves' disease. Iatrogenic means due to the injection of drugs, for example, or chronic hypoglycemia, inflammation, gene mutation. So there is a wide variety of things that may cause endocrine dysfunctions, and you will learn about them more and more during the module and later during internal medicine rotations. Thank you. If you have any question, let me know.